Hey guys, today we are going to finish up meiosis and we're going to talk about meiosis and variation. So there are three sources of genetic variation from sexual reproduction. The first is the independent assortment of chromosomes during meiosis, the second is random fertilization, and then the third source is crossing over. So independent assortment. We have 23 pairs of chromosomes in humans, so a total of 46, but it's divided up into 23 pairs. One half of that pair is our maternal or chromosome from our mom, and then the other one is paternal or chromosome from our dad. So as our gametes are being made, the chance that we pass on either the maternal or the paternal of each pair is one half. So let's take a look at a quick diagram for what that means. So if we are looking at this example where we have two pairs of chromosomes, there's two different probable arrangements of chromosomes at metaphase 1. So possibility 1 would be that we have both paternal chromosomes on one side and both maternal chromosomes on the other side. Possibility 2 would then be that we've got a paternal and a maternal on one side, and then vice versa, the other maternal and paternal on the other side. Then during metaphase 2, you end up with these four different possibilities. So if we are looking at possibility 1 for metaphase 1, when we get to metaphase 2, this is what we are left with. And so we have our combinations down here for possible gametes. If we look at this side, if we're over at the right now with metaphase 2, now you can see that we've got one paternal and one maternal, and so we're going to end up with one paternal and one maternal in these combinations as well. And I know I'm blocking one combination back here, but it looks just like this one. So gamete possibilities, since we have 23 pairs of chromosomes, the number of combination types, it's going to be pretty high. So it's going to be 2 to the 23rd, or over 8,388,000. So we have lots of different possibilities here when we talk about how many chromosome pairs we actually have. Next is random fertilization. So which sperm fuses with which egg is completely random. So females will release an egg. They have no control over which egg is released, and so it's random every cycle. And then lots and lots of sperm, as you can see here. So this is all sperm and on top of an egg. Lots of sperm are going to try to actually penetrate that egg. It's going to be random which sperm fuses with that egg first. So if we have over 8 million kinds of sperm and over 8 million kinds of eggs, then the number of possible combinations of offspring is actually over 64 million. So this random fertilization really increases genetic variation. So is it really a wonder that you maybe look pretty similar to your parents, but you're not exactly like them? Or if you have siblings, that you look pretty similar to your siblings but are not identical to them. Finally, we have crossing over. I kind of referenced crossing over a little bit in an earlier lecture. I talked about the chiasmata, and that is when crossing over happens. So this is going to be the exchange of sister chromatid material during synapsis, and it occurs only during prophase 1. The chiasmata is the point of contact where two chromosomes are crossing over, so I referenced that in the last lecture, but we didn't go into a ton of detail. Here we are looking at prophase 1 of meiosis. This is the only time that crossing over occurs, and we have a pair of homologous chromosomes. And so the two blues, those are the paternal sister chromatids. Then the two reds, those are the maternal sister chromatids. Those are going to form the tetrad. When they're connected like this, you can see we've got these areas, the chiasmata, where part of the blue and part of the red are connected, and they're actually going to switch genetic material. It's a little bit easier to see in this diagram. So again, we have our tetrad. So we have our paternal sister chromatids, our maternal sister chromatids. They're forming this tetrad. While they're closely associated like this, there are going to be areas where these chromosomes actually link up and switch little chunks of genetic material. So what that means is if we're talking about chromosome 1, so this would be the paternal version of chromosome 1, the maternal version of chromosome 1. these two chromosome ones, although they are different chromosomes, so like one is from mom, one is from dad, they contain the genes for the same information. And the genes are located in about the same spot. So if they kind of break off and switch material, all the genetic information that's needed is still there and it's still in the correct spot. You're just creating a new combination. And so again, this happens during meiosis 1, during prophase 1. Now you can see that once we separate them, you've got mostly blue sister chromatids, but you've got a little bit of red. And then you've got mostly red sister chromatids with a little bit of blue. And so again, we have increased this genetic variation because now when we look down here at the possible gametes, this one is just the same paternal chromosome as before, but now this is a recombinant chromosome. So it's a little bit different than what was there originally. Same thing here. So this is another recombinant chromosome, and then this one is the same maternal chromosome as before. So we've now increased genetic variation because we've come up with some new combinations. So why is this important? It's going to break old linkage groups and create new linkage groups, and so again increasing genetic variation. However, this is really common during meiosis, but it's going to be more common towards those far ends of the chromosomes. So with crossing over, offspring can never be 100% like a parent if sexual reproduction is used, because it's really common to have multiple crossing overs, especially on these really large chromosomes. But keep in mind that those crossing overs are going to be more common the closer to the ends of the chromosome. As you get closer to the centromere, you're not going to see crossing over occur as often. All right, so things that you guys need to know for this chapter on meiosis. You need to know how the chromosomes separate during meiosis. So you need to know meiosis 1 and 2 and all the steps. You need to know how meiosis differs from mitosis. And you also need to know how sexual reproduction increases genetic variation, which is what we just talked about today. So your lecture question is, please explain to me why variation is increased with meiosis, but not mitosis. So again, please don't forget to send me a message on the class website with your answer. All right, guys, thank you.